Hello, my name is William, and in this video, I will explain some of the biological mechanisms behind ovarian cancer, how it can begin, develop, and spread. Then, at the end, I will highlight some resources that explain the topics I will cover in greater depth. I hope this video can be a jumping off point for you, so that you can begin your own research on ovarian cancer biology with more confidence from a stronger understanding of the basics. In some people, ovarian cancer begins with an inherited mutation of a predisposition gene such as BRCA1 or BRCA2. These are known hereditary cancer predisposition syndromes, where just one mutated copy of the gene passed down by one parent can significantly increase the risk of developing ovarian cancer if the other copy, with normal function, is lost as a second hit in an ovarian cell. The second hit is commonly a loss of heterozygosity event, in which a region of the chromosome that contains the normal copy of the gene is lost as part of the larger chromosomal changes that occur when cell division is disordered. The normal function of BRCA1 and BRCA2 is to repair damaged DNA, but if mutated, impaired proteins will be produced and an essential mechanism to repair certain types of damaged DNA is lost. The consequence is a profound increase in new mutations with each subsequent generation of cell division. Other suppressor genes are less commonly the initial genetic driver of ovarian cancer, but are commonly acquired new mutations as the cancer develops. Those include TP53, NF1, TERT, NOTCH3, and MYC. Mutations are not the only way ovarian cancer can begin. Changes in how genes are read when activated and transcribed into RNA can also bring about events leading to ovarian cancer. Our genes are transcribed or read by enzymes that copy the DNA and turn it into RNA, shorter strands that can leave the nucleus of the cell and enter the endoplasmic reticulum, where ribosomes translate or assemble messenger mRNA into amino acids, which then assemble through enzymatic action to form the proteins that perform the functional roles in keeping the body running properly. The coiling of DNA around its histone protein backbone affects the type and frequency of proteins made. In other words, the tighter the DNA, the harder it is to read that section of DNA, and therefore, the harder it is to make the proteins associated with that section of DNA. Molecular tags like acetyl and methyl groups attached to the beginning of a genetic sequence increase and decrease respectively the transcription of each gene. These are epigenetic factors that coordinate different gene expression levels and, therefore, the number and type of proteins that are produced in different cells. Of course, disruption of the delicate balance of proteins can contribute to cancer. Examples include hypomethylation of oncogenes, i.e., insufficient silencing of genes associated with processes that can turn cells cancerous, or hypermethylation of tumor suppressor genes, i.e., too much silencing of genes involved in processes that prevent cells from becoming cancerous. Therefore, both genetic mutations and epigenetic factors can contribute to the formation of cancer and the biology of an existing cancer. Once a cell is cancerous, it doesn't behave like a normal cell. It ignores the signals that tell it to stop growing. It replicates uncontrollably, and its genes become less similar to healthy cells. There are a number of so-called hallmarks of cancer that mark critical steps of cancer development, including sustaining proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing angiogenesis, and activating invasion and metastasis. All these factors turn cancer cells into a force that requires much more energy, depriving the rest of the body of vital resources. All forms of ovarian cancer, no matter the location or severity, contain a cascade of changes to the genetic code of cells, and that influences the essential functions of those cells. Genetic mutations may activate critical processes, such as during the S phase of the cell cycle, the time when a cell synthesizes new DNA to duplicate its genes in order to divide into two cells. In one example, mutations activate cyclin proteins to form a complex with cyclin-dependent kinases that will activate proteins and promote cells to enter the S phase, pushing the cell to divide. Critically, cells also have a natural defense system of tumor suppressor genes that tightly regulate critical functions like cell division. Without functional suppressor proteins, cells will be free to grow larger and divide uncontrollably. The retinoblastoma protein is an important tumor suppressor that prevents the cell from progressing through the cell cycle if there is faulty or unnecessary division. So, the cascade of genetic aberrations in ovarian cancers can combine to activate key cellular functions and disrupt their usual regulation, which leads to uncontrolled growth. The patient's own normal cells can also influence ovarian cancer biology. Normal immune cells, like cytotoxic CD8 positive T cells, can protect against cancer cells because they can recognize and destroy cells that do not have the correct identifying molecular markers on their external surface. These immune cells can use these markers to distinguish between the body's healthy cells and foreign cells, such as virally infected or cancerous cells. The immune system can then signal the foreign cell to commit apoptosis, or programmed cell death, i.e., cell suicide. But if the cancer cell goes unnoticed or is resistant to the immune system, the cell will avoid this defense and continue to accrue mutations and evolve as a cancer. Tumors are masses containing cancer cells, but also other non-cancerous cells. In fact, tumors are commonly referred to as a microenvironment because immune cells and markers, blood or lymph vessels, connective tissue cells, healthy cells, and vesicles containing nutrients are all present too. Cancer cells utilize this microenvironment as a support system, protecting them from the body's innate cancer defense system and funneling them the resources they need to grow and spread throughout the body. As ovarian cancer grows, it also develops the ability to spread to other sites and uses the microenvironment to infiltrate and grow. Ovarian cancer looks and behaves differently from these other cancers, in part because the embryological code for development into an ovary is still programmed into the biology of the cancer cells. Just as individual cells of an embryo differentiate into mature tissues and organs, cancer cells can use embryological biology to change their properties over time. Again, normal biology is subverted by the uncontrolled and altered proteins in the cell. In a process called epithelial mesenchymal transition, or EMT, cancer cells revert to mesenchyme when they downregulate, produce less E-cadherin, a critical molecule for intercellular connection that helps epithelial cells stick in an organized mosaic with their neighbors. Thus, the cancer cell, now more round in appearance, reverts from a specialized epithelial arrangement that normally covers the ovaries back toward a pluripotent mesenchymal stem cell, which confers the ability to evolve more variably, survive adversity, and more easily infiltrate and spread through tissues. Cancerous stem cells can also circulate until they settle onto an area of tissue. At a distant site, mesenchymal cancer cells can lodge into the tissue through the opposite process of mesenchymal epithelial transition, MET. They upregulate E-cadherin again and infiltrate the tissue by presenting the protein FOS on their cell surface that binds to the FOS receptor on the surface of the mesothelial cells, clearing the way for the tumor to invade and grow into the tissue. The area it left is called the primary site, and the area it colonized is called the secondary site. There can be multiple secondary sites, depending on the extent of metastasis. The most common sites of metastasis in ovarian cancer are the peritoneum, lymph nodes, lungs, liver, and on rare occasions, the bones and brain. Low-grade mucinous ovarian cancer begins in the peritoneal space of the abdomen, the location of the outer surface of the ovaries. The passive movement of peritoneal fluid aids in the metastasis of ovarian cancer cells through the peritoneal cavity. In contrast, the more common high-grade serous ovarian cancer originates in the fallopian tubes and universally carries a TP53 mutation, lying dormant for decades while shifting from abnormal tissue to a serous tubal intraepithelial carcinoma to a high-grade serous carcinoma. Genetic predispositions accelerate the development of high-grade serous ovarian cancer, a finding that has prompted a call for detection through cervical liquid sampling and prophylactic prevention through surgical means. Often the first step of overall treatment is surgery, which aims to physically remove as much of the tumor as possible, and it is shown that doing so improves survival in patients with ovarian cancer. The next step is usually chemotherapy, which is a therapeutic poison to the body that works by damaging the DNA of replicating cells. Healthy cells exist in a stage of the cell cycle in which division is unnecessary, but cancer cells rapidly grow and replicate, and are therefore much more likely to accumulate and succumb to the genetic damage from chemotherapy. Another common treatment is radiation therapy, which works by damaging DNA in rapidly replicating cells using high-energy particles or waves. These beams are precisely aimed at tumors to inflict enough damage to cancer cells that they are unable to repair themselves. Then there are PARP inhibitors, which take advantage of the impairment to DNA restoration conferred by the BRCA1 and BRCA2 mutations acquired by cancer cells. This treatment inhibits the PARP protein from repairing cancerous cells after DNA damage. Other promising therapies used to treat ovarian cancer are immunotherapy and the use of tyrosine kinase inhibitors. Immunotherapy, also known as immune checkpoint inhibition, prevents the body's regulatory immune cells from restricting the anti-cancer response so that the immune T cells are free to carry out their attacks against cancer cells with abnormal markers. Tyrosine kinase inhibitors block the tyrosine kinase proteins, which are overexpressed in cancer cells, from signaling growth and division. These contemporary treatments all seek to eliminate ovarian cancer by exploiting a biological characteristic of the disease. Your physician will design your personal treatment plan with you, but it is also worth mentioning the importance of researching clinical trials that may be available to you. Clinicaltrials.gov is a website the US government provides that shows all available clinical trials in the world. By searching for a disease like ovarian cancer, a drug name, or any other relevant search term, you can find a list of clinical trials recruiting patients that fit their criteria. Studies show that patients with commonly occurring cancers enrolled in clinical trials have a lower risk of adverse outcomes. Indeed, clinical trials usually compare the current best treatment with promising new treatments that have the potential to improve on that. For a more targeted search, you can use the ovarian cancer-specific clinical trials search tool on Overcome's website. I sincerely hope this video explaining the basics of the biology behind ovarian cancer has been helpful. You can find more in-depth information here from current scientific papers and reviews, and of course on Overcome's website, which has information on ovarian cancer prevention and awareness, testimonials from patients and renowned experts, and ways to get involved in the fight against ovarian cancer. Thank you. I wish you the best in your research journey.